Thank you for the invitation to come and speak. Please let me know if you can't hear me. I'll try and speak up. Okay. So an outline of the lecture is, number one, we're going to talk about DNA and where we get it from. We're going to talk about early development after normal fertilization. And we're going to talk about reproductive technologies to prevent disease, and then reproductive technologies to treat disease. So DNA is the substance of inheritance. It's organized into genes which encode proteins, and the proteins, of course, are the building blocks of all our cells. Now, in the cell, we have two types of DNA. We have DNA in the nucleus, which is packaged into these large linear molecules called chromosomes. And then we've also got DNA in the mitochondria, and that's packaged into a structure called a nucleoid, and it's a circular molecule of DNA. Now, the mitochondria are organelles in our cells, and they're unique in containing their own DNA. So why do they have their own DNA in the first place? How do they come to have it? So the story goes that about 2 billion years ago, precursors of our eukaryotic cells engulfed this bacterial ancestor of the mitochondria. And it turned out that the mitochondria could produce energy in the form of ATP, which fuels 90% of our cells' energy requirements. And this has been described as the most important meal in history. So mitochondrial DNA is a remnant of the bacterial ancestor's genome. And here's a schematic of a mitochondrion. It's a double membrane structure, and here on this inner membrane is where it forms the ATP, which is the energy it supplies for the cells. And within its lumen here, it's got its circular molecules of DNA, which circular like all bacterial genomes. And the mitochondrial DNA encodes just 37 genes. 13 of them are proteins, all involved in energy production, which you see in the next slide. And it encodes 2 ribosomal RNAs and 22 tRNAs. Now, it takes a lot more proteins. It takes an estimated 11 or 1,200 proteins to make the mitochondria work. But during evolution, they have outsourced most of this to the nucleus, and they just hold on to this small number of genes. So our nuclear DNA encodes about 37,000 genes, and only 20 to 25% of them encode proteins. So the nuclear DNA encodes what we call our sort of identifying traits, and most of the other proteins required for the cells to function. Proteins encoded by the mitochondrial DNA are involved only with energy production. I showed you this energy factory here consisting of 5 protein complexes on the inner membrane. Now, they're predominantly encoded by nuclear proteins, shown in the blue, and in the darker color you see the mitochondrial proteins. So within each of these protein complexes, we've got proteins encoded by the mitochondrial DNA. And so the nuclear and mitochondrial encoded proteins work together to supply our cells energy. Now, if we get mutations in either the nuclear or the mitochondrial DNA, we can get serious disease. And DNA mutations, as I'm sure you know, they're like spelling mistakes, and if those spelling mistakes are bad enough, then we affect protein structure, which can lead to serious disease. And mutations in the nuclear DNA have a wide range of adverse effects on human health. Mutations in the mitochondrial DNA affect mostly tissues requiring a lot of energy to work, such as the brain, the heart, or the skeletal muscle. So we have nuclear and mitochondrial DNA. Where do we get it from? And this is a human fertilized egg. This is how we all start our lives. Here's the fertilized egg, and in these 2 structures we have the pronuclei, which separately contain the DNA we inherit from our mom, from our oocyte, and from our dad, the sperm. And each of those contains 23 chromosomes, just a single copy of each. So when this embryo begins to divide, these combine so that we get a copy from mom and a copy from dad, and each cell will contain 46 chromosomes. And from this single cell, we get a baby, and 9 months later, which is quite staggering by anybody's measure. So nuclear DNA is inherited from both parents, but the mitochondrial DNA is inherited only from our mothers. So the sperm does have mitochondria. It needs it to have the energy to swim all that distance. But once fertilized, and you can see that here in this nice graphic, the mitochondria are in the neck of the sperm, but once it gets into the egg, they're killed. They don't persist. So the sperm mitochondrial DNA does not persist after fertilization, and the nuclear DNA contained in each zygote is unique, and mitochondrial DNA is the same as mom's because we just inherited from the egg. So why are we not identical? It's important to understand this point in relation to how we view cloning, for example. So as we mentioned in the last slide, every fertilized egg has a unique nuclear genetic composition. How does this come about? A specialized form of cell division halves the chromosome number in preparation for fertilization, and it also creates genetic diversity. So in a normal cell division, where any of our cells, how they divide, including the embryonic cells, you've got one chromosome each from mom and one from dad. It replicates its DNA, and then when the cell is ready to divide, these chromosomes align on a spindle and they're pulled apart. And so you get a copy from mom and a copy from dad going to each daughter cell. And in this way, mitosis enables dividing cells to retain 2 copies of each chromosome. Meiosis, on the other hand, it's got to halve the chromosome number. And it does this by bringing mom and dad chromosomes together, and they form linkages here during meiotic recombination. So they're physically attached. And then during the first meiotic division, these whole chromosomes separate. So instead of having one sister going to each spindle pole, now we've got a pair of sisters going to each spindle pole. And what you notice is that those chromosomes are a mix of mom and dad, and that has happened during meiotic recombination. So this is the basis of how we reduce to a single copy of each chromosome and how we create genetic diversity. This is the basis of our genetic uniqueness. During the second meiotic division then, it's more like mitosis, where you get these recombinant chromosomes dividing and being pulled to opposite sides of the poles, and what you get from most meiosis is 4 single haploid nuclei. That would happen in sperm and most other organisms. However, in female meiosis, we have only one viable haploid nucleus. And so if we look at the egg here we have the first meiotic division, where these homologous chromosomes joined together are being pulled apart. Then we have the second meiosis, where it waits to be fertilized, and then completes meiosis following fertilization. And what happens is that most of the chromosomes, in the first meiotic division, we lose half of the DNA in the first polar body, this tiny inviable structure here. In this second meiotic division, we lose the other half of the DNA in the second polar body. So during female meiosis, 75% of the genetic material we inherit from our parents will be lost. Only 25% is retained in the oocyte, and it's purely stochastic. It's going to be a mix of genes from each of our 4 grandparents. So meiosis enables sperm and oocytes to transmit just 1 copy of each chromosome, and importantly creates genetic diversity. So this is just taking a closer look at these oocytes. So this is an immature oocyte. Here it's got 46 chromosomes. Then it's got to halve these during these meiotic divisions. This is the polar body here in a human oocyte, the first polar body. And then the first polar body, this is the second polar body. First polar body normally degenerates. This is going from an immature oocyte, which then undergoes meiosis and forms a fertilized egg. And the chromosomes are doing, as I said in the last slide, you've got mom and dad joined together. Then they come apart during the first meiotic division. And then during the second meiotic division, the sister chromatids that are left in there come apart, leaving you 1 copy in the nucleus, in the pronucleus. And of course, you've got a single copy in the male pronucleus. So key points about the inheritance of our DNA. Meiosis generates exactly 1 copy of each chromosome. Nuclear DNA is transmitted by the sperm and egg and contains a random mix of genes from each of our 4 grandparents. This gives each of us a unique genetic identity. Mitochondrial DNA, on the other hand, is invariably inherited from our maternal grandmother. And during meiosis, the oocyte loses 75% of its nuclear DNA, but retains virtually all of the mitochondrial DNA. So during pre-implantation development here, we've got the fertilized egg dividing to 2-cell, 4-cell, 8-cell, then it forms a stage called a morula, which isn't shown here, and then it develops to the blastocyst stage. Blastocyst stage is a really significant stage because the embryo has to develop to this stage before it can implant in the uterus. And I'll talk more about that in the next slide. So during this development, during each round of cell division here, the nuclear mitochondrial DNA or the nuclear DNA is replicated, such that by down here we have more than 200 copies of nuclear DNA. However, the mitochondrial DNA shows no net increase according to our current understanding. So all of the mitochondrial DNA contained in the oocyte, and it is between 200,000 and 500,000 copies, will just partition between cells so that by the time you get down here, you have relatively low numbers of mitochondrial DNA in each cell. So just to take a closer look at the mammalian blastocyst here, it's composed of an outer layer of trophectoderm cells and an inner cell mass. So the trophectoderm gives rise to a placenta, the inner cell mass gives rise to a primitive endoderm and the epiblast. The primitive endoderm goes to the yolk sac, and the epiblast gives rise to fetal tissues if this embryo implants. Or if we culture this inner cell mass in vitro, we can get embryonic stem cells. Now, an important point is that this is a human blastocyst stained for a protein called nanog, which is expressed in epiblast cells. So what you see here is a tiny population of cells that give rise to fetal tissues. So the whole emphasis of development during the pre-implantation stage is on the production of extra embryonic tissue. Only a tiny number will go to form fetal tissue. And just to emphasize that, here's another blastocyst, and this time I'm showing you another fixed blastocyst with its epiblast precursors expressing nanog. And what I want you to see here is in the hatched blastocyst, which is hatched from its outer shell and it's ready to implant, that contains a very small number of epiblast cells, placed between 5 and 12 epiblast cells, as you see here. So these babies are derived from a tiny founder cell population, and that gives rise to all of the cell types in the human body. It gives rise to the mesoderm, which gives us our skeleton, muscular system, you can see them all there, and the endoderm and the ectoderm, which is the nervous system and the skin. It also gives rise to, you have got these pluripotent nanog-expressing cells, then become differentiated to all different cell types in the body, and that happens just after implantation. Now, the founder cells in the blastocyst also give rise to germ cells, and I'm going to focus here really on oocyte development because this is the one who sort of transmits the pluripotency from one generation to another. So, about 3 to 5 weeks of gestation, you have these cells migrating to the gonadal ridge to form, in the case of females, to form the ovary. And then this relatively small number of cells that migrates divides rapidly to give you about 7 million oocytes. And by birth, we have about 1 million of these. 1 million of these are left. So in germ cell development, we've got pluripotent cells become differentiated because germ cells are highly differentiated. However, they have the capacity to return to pluripotency. In case I didn't explain that very well enough, the pluripotent cells are the ones that have the capacity to develop into every cell type in the body. Now, having covered normal development, we're going to talk about assisted reproductive technologies to prevent transmission of disease due to inherited mutations in nuclear or mitochondrial DNA. So, as we heard earlier, if we have mutations in the nuclear DNA or the mitochondrial DNA, there is a risk of serious disease. Sorry, we saw this earlier. And these nuclear DNA mutations are inherited from both parents, and mutations in mitochondrial DNA are inherited just from our mothers. So a current option for reducing risk of transmitting disease is pre-implantation genetic diagnosis. I'll tell you more on the next slide, but this is a well-established technique for preventing transmission of nuclear DNA mutations, and it's now offered by a limited number of centres to reduce the risk of transmitting mitochondrial disease. So for a couple who are at risk of transmitting a nuclear defect, they undergo IVF treatment to produce multiple embryos. Then we remove either 1 or 2 cells from this 8-cell embryo, or we wait until it develops to the blastocyst stage and we remove a number of cells from here. And that the embryo will still be capable of implanting following removal of these cells. But these cells can be then sent to the genetics lab for testing. And if the gene defect is present, we don't replace that embryo. But if it's absent, we do. So we select disease or mutation-free embryos for replacement. So PGD for nuclear mitochondrial mutations is fairly straightforward. For PGD for nuclear DNA mutations is fairly straightforward. But for mitochondrial DNA, it's complicated, a bit more complicated. It's complicated mainly by the fact that these mutations can be present in either some or all copies of the mitochondrial DNA. And the severity of disease depends upon the mutation load. So if we look at a woman's eggs here, there are 3 options. She either has no mutation, it's all wild-type mitochondria, in which case you get a normal embryo. She's got a mix of wild-type and mutated mitochondria, in which case we get an embryo with a mix of these and a baby with a mix of these, and the severity of disease depends on how much mutated there is relative to wild-type. Another option is that 100% of the mitochondrial DNA are mutated. Now, for a woman in this situation, all of her children will inherit the mutation, and all of their mitochondrial DNA will carry the mutation. So there is a very high risk of serious disease in these cases. But the actual severity of, even with 100% mutation, the severity of disease varies between individuals. And here, the severity of disease depends again on the mutation load. So when there's 100% mutation, we call that condition homoplasmy. All oocytes carry 100% mutation load. For women who are heteroplasmic, there is wide variation in mitochondrial DNA mutation load between oocytes. So if we go back to our PGD here for reducing the risk of mitochondrial DNA mutations, we can take a single cell from this, or 2 cells, and use that to predict the mutation load in the entire embryo. Again, we do the same thing. We have IVF treatment to generate multiple embryos. We remove cells from an 8-cell embryo or from a blastocyst stage embryo, and embryos with high mutation loads are excluded from use in treatment. So the summary of PGD for mitochondrial disease, the evidence so far, although it's pretty early days with this treatment, so far indicates that PGD provides a promising risk reduction strategy for affected families. However, it is not useful for women who are homoplasmic from mutations or whose embryos consistently carry high mutation loads. So what can we do for these women? So one strategy we've been pursuing is to see whether we can uncouple the inheritance of nuclear DNA from mitochondrial DNA, to see whether a woman could have a genetically related child without the risk of transmitting disease. And because there are so many mitochondria in the oocyte, it's not feasible to go in and pick them all up. So the most realistic thing to do is actually transplant the nuclear DNA between eggs or zygotes. So the concept is that by this nuclear transplantation, you would create a reconstituted zygote, that's a fertilized egg, containing nuclear DNA from the affected woman and her partner, and mitochondrial DNA from a healthy donor. And here, because when we're taking these mitochondria, or taking these pronuclei, we pinch them off in a piece of cytoplasm, so they will also contain some mitochondria. So there will be a small amount of mitochondria from the affected woman. Okay, so the current options for the transplantation of nuclear DNA, there are 3 options currently on the table. The first is pronuclear transfer. So that is, you get this egg and this is, I'm showing this in the context of how it would be if we were doing clinical treatment. But I should stress there are no clinical treatments currently being done with these procedures because it's not currently legal to do so. So we've got an oocyte donated by one woman, and then we've got a patient oocyte. So we fertilize them both. We get a fertilized egg. Then we take the pronuclei, the nuclear DNA, out of the donor oocyte, remove it. And what we do is we remove the pronuclei from the patient oocyte also, and then fuse it with the donor oocyte, the enucleated donor oocyte, and in this way we get a reconstituted zygote. Another option for this is metaphase 2 spindle transfer, and this is the metaphase 2 oocyte here. And one of the issues here is that you can't actually see, because the chromosomes are arranged on a spindle, and it's not in a nucleus at this stage, is you can't actually see what you're doing by ordinary light microscopy. So we use a special microscope to see the spindle, which you can just see here is flickering, but you can't actually see the chromosomes. So again, the concept is the same. We take the spindle from the donor oocyte, we discard it. We take the spindle from the patient oocyte, and then we fuse it with the enucleated donor oocyte. We fertilize it, and then we get a reconstituted zygote. And something that's been more recently proposed is to use this polar body, because if you remember from the meiosis slide, this contains half of the chromosomes from the mother. And the idea is that if you could also use that to fuse it with a donor egg, then we would be able to get 2 genomes from each patient oocyte, potentially, and it would reduce the cost and reduce the inconvenience to women who are affected by mitochondrial mutations. So just to go through where we are with these, we've got this pronuclear transfer was first done in the mouse and published in Science in 1983. We did proof of concept work published in 2010, and I'll talk about that in more detail in the next slides. Then the metaphase 2 spindle transfer proof of concept first came from work in the monkey in 2009, and then in the human 2012 from 2 groups in the US. This polar body transfer has so far just been done in mouse, and that was published by a Chinese group last year. It hasn't been tested in human yet. So all of these approaches give us the same outcome. They give us a reconstituted zygote containing mitochondrial DNA from the healthy donor, nuclear DNA from the affected woman and her partner. So I'm going to focus more on pronuclear transfer, because that's the approach we've been pursuing in Newcastle. So it was first published in the mouse in 1983, and then there were multiple subsequent studies. And then later in 2005, a Japanese group actually generated a mouse model with a deletion in its mitochondrial DNA and showed that they could vastly reduce the persistence of that deletion to offspring when they performed pronuclear transfer. And then in 2010, there was proof of concept using abnormally fertilized zygotes from work that we did. Now, I should say the reason we did the proof of concept work on the abnormally fertilized zygotes is that we work in an IVF lab, and patients, of course, want to keep their normally fertilized eggs for their own treatment. So this was the source of material that was available to us when we started. And so but using this source, we were able to figure out ways of removing these pronuclei, which in the human are very large structures. And removal of the pronuclei involves the use of chemical inhibitors to depolymerize the actin and microtubule network. So all of our cells have this kind of cytoskeleton that gives them their shape and form. But in order to go in there and suck out that pronucleus, we need to disable that. And that can be done reversibly using chemical inhibitors. And then the next step is to fuse this pronucleus back with an enucleated cytoplast. So this piece of this pronucleus surrounded by a piece of cytoplasm is called the karyoplast, and this enucleated zygote is often called the cytoplast. So fusion of these was a big struggle for us, because the convention at that time was to use an electrical chamber, but human oocytes didn't tolerate that well. So we went back to a yet older way of doing it, in which they use a technology from the virus. So an inactivated virus still has its viral envelope protein, which allows it to fuse with cells. So by just bathing this karyoplast in a solution of the viral envelope protein and then placing it next to the plasma membrane, it fuses on its own, and that turned out to be a big breakthrough in our step towards making this work. So then we could take them in and take the pronuclei out and get them back in. The question is, would they develop to blastocyst stage? So for our unmanipulated controls, we had 17% development of blastocyst, and that's much lower than normal. For normally fertilized eggs, we get about 50% development of blastocyst stage, but because these are abnormally fertilized, they had a low capacity for onward development. But with our pronuclear transfer, we got 8% development of blastocyst stage. So there's about a 50% drop-off. But nonetheless, we thought that was enough of an indication to say we should investigate this a bit further. And of course, the other important question here was to ask how much mitochondrial DNA was persisting in this karyoplast. Because if we're getting large amounts of mitochondrial DNA from the patients here, it's not going to be very effective as a treatment. So and you can distinguish between the two mitochondrial DNAs from the patient and the donor by identifying sequence variants in this D loop, this area of the mitochondrial genome that's highly variable. So using this approach, we were able to measure the amount of mitochondrial DNA that we carried over. And after some optimization of the procedure, we brought it down to about 2%, which is way below the level for disease or disease transmission. So in summary, for the proof of concept for pronuclear transfer in the humans, we found it's technically feasible to do the enucleation and fusion. We found it's compatible to the blastocyst stage. They were humble beginnings, but we got some blastocyst development, and we found that the levels of mitochondrial DNA were carryover were sufficiently low to make us confident that the disease would be not transmitted to individuals undergoing this procedure. However, meaningful tests on the safety and efficacy of pronuclear transfer, because if we're going to take this to treatment, we've got to be happy that it's going to work, or that there's a reasonable chance of working, and that it's going to be without any known harmful effect. So in order to test that, we needed a supply of normally fertilized zygotes, and this makes things a bit difficult. So where do we get these oocytes for research? We get them from women undergoing IVF treatment who share their eggs for treatment who share their eggs for research in return for reimbursement or subsidy from the research fund. Alternatively, we get altruistic egg donation. Donors are reimbursed for their expenses in this scheme. However, only 5.5% of women who express interest in altruistic donation eventually donate their oocytes. So in our ongoing work here, we're aiming to further refine the technique to maximize development of the blastocyst stage. And we want to compare pronuclear transfer blastocyst with unmanipulated controls. And here we've got an image of a control day 5 blastocyst, and then it develops on day 6, it expands. And then we've got a pronuclear transfer blastocyst, and which expands, and because we have to make a hole in the zona, they tend to escape out their zona, but that's the same as what happens for PGD embryos. So morphologically, these embryos are very similar, and our ongoing studies are looking at whether these blastocysts are normal or not. So we've developed a system whereby we can really get a lot of information now from individual blastocysts. We can take individual cells from the inner cell mass. We can do gene expression analysis and compare them with controls. We can do, sorry, I've lost something here. We can do aneuploidy screening. So that is asking, do they have the correct number of chromosomes? And then we can do analysis of mitochondrial DNA carryover. So from a single blastocyst, we can get a lot of information, and we're making very good progress with that at this point. So next, I'm going to talk about the other option on the table. That's metaphase 2 spindle transfer, and that's predominantly the work of these two groups in the US. So, as we mentioned, this metaphase 2 spindle can be visualized by using polarized light, and then it can be removed and transplanted. So in 2009, we had a publication in Nature from a group in Oregon where they got live monkeys following spindle transfer in monkey oocytes. These live monkeys are now 6 years old and they appear to be normal. And then in 2012, they followed that up with proof of concept in human. So this is the metaphase 2 spindle in the human, and then they take it out and fuse it with another oocyte. But what they found was they got a very high incidence of abnormal fertilization. So normally fertilized zygotes, as we saw, should have two pronuclei. Here they got a high incidence of either 3 or 1, and only 48% fertilized normally. So this is a limitation to the efficiency of metaphase 2 spindle transfer, but there is some confidence that this problem will be sorted out. And the thinking is that this is a problem. This abnormal fertilization is due to a problem with the extrusion of this second polar body. So the oocyte retains it and has an extra copy of each chromosome. But the point is, you can see that. That's obvious when you check the fertilization, so we can exclude those from treatment. And of those then, despite this abnormal fertilization, this group reported a high development of blastocyst stage. 30% of embryos developed a blastocyst stage compared with 50% of controls. So, to summarize where we're up to with these techniques for preventing transmission of mitochondrial DNA disease, we've got pronuclear transfer and spindle transfer provide a promising approach to reducing the risk of mitochondrial DNA disease. Further studies are required to test the likely efficacy and safety before we can go to treatment. And then we've got polar body transfer, which is interesting but has not yet been tested in human oocytes. So that was how we can use our technologies to prevent transmission of disease. But assisted reproductive technologies can also be used to treat disease, potentially. So in this slide, I told you that these tiny population of epiblasts, these pluripotent founder cells in the blastocyst, can either form fetal tissues if they implant in the uterus, or can form embryonic stem cells if they're cultured in vitro. So by culturing, what the aim of culturing ES cells is that we want to harness the state of pluripotency. Now in the embryo, pluripotency is really transient. It's there in the blastocyst, and then by the time it implants, it's lost. So by explanting the inner cell mass, which you'll see here, the intention is to harness pluripotency. So we cut the inner cell mass out, place it on these feeder cells, which then form these outgrowths. So this is an inner cell mass that's just plating down on the feeder cells. And then it forms colonies, multiple colonies of ES cells. And here's a close-up of this. This is typical human ES cell colony, just full of cells in there. So all of these cells will be pluripotent and therefore have the capacity to generate, to develop into other cell types, and can potentially be used to treat degenerative disease where there's tissue or organ failure. So embryonic stem cells are derived from pluripotent cells in the early embryo. They were first derived in the inner cell mass of the mouse blastocyst, 1981. That was published in 1981. The first human embryonic stem cells were published in 1998. And at this stage, there are hundreds and hundreds of ES cell lines, and they're typically derived 5 to 7 days after fertilization when the embryos reach the blastocyst stage. So why are we interested in these ES cells? They have therapeutic potential. As I mentioned, they can be used to generate cells from almost any cell type for transplantation. They can be used for the production of transgenic mice. They can be used to study developmental biology, to study how the cells in the embryo assign themselves to one cell fate or another. They can be used for toxicology study, and they can be used as disease models if we can derive ES cells from genetically abnormal embryos. So there are lots of reasons to be interested in this. However, for therapeutic potential, there is a limitation, because the patient's immune system might reject these stem cells, And this problem could potentially be overcome by making ES cells with the same genetic constitution as the patient. So this is the driver for the deriving what we call isogenic, same genetic constitution as the patient, isogenic ESCs. How can this be achieved? So the question is, can the process of differentiation be reversed? When I was showing you embryonic development, I showed you how the small pluripotent population formed all the cells in the body through differentiation around the time of implantation. So in order to get an isogenic cell line, we're going to ask whether a differentiated cell can be returned to the state of pluripotency. So the idea of using oocytes to reverse differentiation is sort of the rationale is based on fertilization. Here we have this highly differentiated sperm cell, fertilizes the egg, and it's reprogrammed, and then can contribute to the forming of cells that can form all cell types in the body. So the oocyte can reverse differentiation if you take the sperm example. So that led to the technique of somatic cell nuclear transfer, which is also called the Dolly technique, used to create Dolly the sheep. So what happens here is that we take an unfertilized egg, we remove the spindle and chromosomes from that, we have an enucleated egg, and then we fuse a somatic cell with that to create a cloned embryo. So the terminology here can be, this can be called cloning, it can be called somatic cell nuclear transfer. For simplicity, it can also be called the Dolly technique. So somatic cell nuclear transfer creates cloned embryos in which the nuclear DNA is identical to the somatic cell, and the mitochondrial DNA is predominantly from the oocyte. So a very brief history of cloning here. It was first reported in 1952 by these two scientists working in the US. They cloned frogs by placing nuclei from an early embryo into enucleated oocytes. And here we see these albino frogs, all clones, derived from the eggs of this wild-type frog. However, these researchers reported in 1957 that only happened before differentiation. If you took a differentiated cell, you could no longer return them to pluripotency. But then in this man, Professor John Gurdon, in 1957, started his PhD in Cambridge and undertook actually to try this again. And so using a different species of frog, he obtained viable offspring following transplantation of epithelial cells from the intestine of a feeding tadpole. Published that in 1962. And the conclusion from that is that the frog egg can reprogram differentiated cells to a state of pluripotency. However, he had no success when adult cells were transplanted. So it could do it, but it was limited to early developmental stages. And you see that here. This title actually comes from, because John Gurdon here won the 2012 Nobel Prize for his work in cloning. And his Nobel lecture was entitled "The Egg and the Nucleus, a Battle for Supremacy." So this graph is showing you the developmental stage from which nuclei were taken. So with advancing development, it is more difficult to reprogram and get normal embryos following this nuclear transfer. So the ability of the egg to actually reprogram, as development proceeds, these somatic cell types get very set in their ways, and it's difficult for the egg to reverse that. This was how it was left. Now we're going to advance forward by 40 years, where we get the first successful live birth from somatic cell nuclear transfer in mammals. And this is Dolly the sheep, smiling broadly here. And here she is with her baby. She had, I think, 5 or 6 lambs. So what I've been talking about in the last couple of slides is reproductive cloning. It was done for the purpose of producing live offspring, and that's valuable for farm animals, for breeding rare species, and so on. But obviously it is illegal to do that in humans. And so in humans, we're looking at therapeutic cloning. We produce blastocysts with the aim of deriving embryonic stem cells. So if we can produce a blastocyst from this somatic cell fusion, then we can get a pluripotent population that has the same genetic constitution as the patient, and we can then use those cells to produce ES cells that have the genetic constitution of the patients and therefore will not be, most likely not be rejected by the patient's immune system. So that is the idea, and it has proved to be rather difficult. So these slides are from work done in my lab. So these are these donor cells we use for somatic cell nuclear transfer. That's their amnion cells that we just get from the amnion at birth of babies, and these are adult skin cells. You can see just these fibroblasts growing flatly on the cell on the tissue culture plastic. And then we do enucleation by going in, making a hole with a laser in the zona. Go in, we remove the spindle, and then we fuse this somatic cell with the egg. So you get a reconstituted egg consisting now of the nucleus of the somatic cell and mostly mitochondria from the donor egg. And then this is activated, and we can trigger onward development of that. And this is showing you the real thing here again. That's the human oocyte. That's the spindle, which we remove. And you can see that spindle there in the pipette. And then this is the somatic cell fusing with the oocyte. So there were extensive attempts at getting ES cells from human oocytes. But almost then in 2013, this was published again by this group in Oregon, where they successfully derived ES cells following somatic cell nuclear transfer in humans. So they grew their fibroblasts, they transplanted it into the metaphase 2 egg, human egg. They got these blastocysts with big inner cell masses, and from these they were able to derive ES cells. So in theory now, we have the building blocks in place to make ES cells that would have the same genotype as a patient who would need treatment. However, meanwhile, this Shinya Yamanaka in Japan, who incidentally shared the Nobel Prize with John Gurdon in 2012, he developed this very simple concept that actually if you forced expression of the genes that are expressed in the embryonic pluripotent cells, then you may be able to convince a cell to become pluripotent itself. So this is called direct reprogramming of somatic cells to a pluripotent state. It's called induced pluripotency. And that was first published in 2006. And remarkably, these cells expressing these, came down to just 4 genes in the end. They can give rise to all somatic cell types. So an intensive area of research at the moment is to see whether there are biological differences between these induced pluripotent cells and cells created by somatic cell nuclear transfer. So just to go back then, before I finish, where do we get our DNA from? To go through the options we've spoken about. At fertilization, the nuclear DNA comes from both parents, and the mitochondrial DNA comes from only mom in the egg. Pronuclear transfer, metaphase 2 spindle transfer, the nuclear DNA comes from both parents, but the mitochondrial DNA comes from the healthy donor's oocyte. And in somatic cell nuclear transfer, we get nuclear DNA from the somatic cell and mitochondrial DNA from the donor egg. And a key point here is that these two have a unique genetic identity, whereas this one, they have a unique nuclear genetic identity, whereas this one is an exact copy of the somatic cell from which it was derived. And with that, I'd like to thank you for your attention and acknowledge the people in my lab who've done the work and our funders, the Wellcome Trust Centre. And also Doug Turnbull's lab, who do the mitochondrial DNA analysis. Thank you.